Hello, I'm Soo Romanoff from Edison Group, and today we're catching up with Bjorn Dahlof and Rahul Agrawal from Cereno Scientific. So Bjorn, you've been the scientific leader at Cereno for a little while, and you've been joined by Rahul Agrawal. I mean, could you give us an idea of how you'll both be working together? And then separately, Rahul, maybe you'd like to share a brief introduction and rationale for joining Cereno.
I think I start then because I've been with the company since 2012. I was almost part of the inauguration of the company. I was brought in to help with, so to say, building a company and medical consultancy. And then I joined the board. I was on the board and I also did consultancy work in parallel with my hospital work. And ultimately, I became the CMO of the company. But then we expanded, and we were not so many people. So I found myself sitting on 3 different chairs, like chief scientific officer, chief medical officer, and head of clinical development. And I've always been very keen on working on the scientific platform and deep into science and try to see the way forward for the company. And it was a little bit too much of operations in the CMO and clinical development role. So I was very happy to see that Rahul came in to the company to, I can see it as unloading me a little, but at the same time, I think we were aim to work together on both the science and the medical. So I will continue to have some CMO roles in a study or something, but Rahul will take the full responsibility for the CMO and the research and development part. But we work together, as said.
Yeah, and thank you so much for asking that question. So by training, I'm a cardiologist and an emergency physician. Did my training in Germany and the US. I've had the pleasure of being in several big pharma companies, Bayer, AstraZeneca, also in senior executive roles as global VP. And before joining Cereno Scientific, also was the chief medical officer and chief development officer at a gene therapy biotech. And I've been privileged also to be involved in R&D as well as commercial parts of pharma business, have been involved in 7 launches, over 30 trials. So what really intrigued me about Cereno Scientific is not only the exciting journey that they have laid down in the last 10 years, you just heard from Bjorn, but also the intriguing compounds that they have. And so it is a privilege for me to join now as the CMO and head of R&D to help bring forward the 3 assets that we have, and very promising assets further in their development.
Great. So what are the opportunities that you identify in utilizing HDACi inhibition as an approach for broader cardiovascular disease? And then more specifically on the pulmonary arterial hypertension?
I mean, I can start since I've been mainly probably involved longer in looking into HDAC inhibition, even though I'm sure that Rahul have been subject to that and looking into those matters also during his other commitments. But I have been going through the literature. That was a survey I did 2019, several hundreds of references showing that HDAC inhibition then being used in cancer indications because of its properties of anti-proliferation, anti-inflammation, and that. I looked into the cardiovascular area and found that there were a whole host of references showing that HDAC inhibition is actually both anti-proliferative, anti-inflammatory, anti-thrombotic, and pressure reducing, and all of those properties made us to rethink that we only had a antithrombotic entity in our company, that we could do more and have a broader cardiovascular approach. And that made, in addition to some other factors, our switch to PAH. But we can come back to that later, maybe.
Yes, so as Bjorn was alluding to, I think HDAC inhibition exhibits some highly relevant properties that are we find desperately needed in the cardiovascular arena. Bjorn was mentioning the antifibrotic, antiproliferative, and also antithrombotic properties, which hold a big promise to address several cardiovascular diseases. We did put our focus on pulmonary hypertension, even though there are some drugs on the market. There has been done some advancement over the past few years, and I had the privilege also being involved in some. Yet most of the activities are more on the vasodilation bit, less so on the remodeling, which is again a big and major part of pulmonary arterial hypertension. Our drug, a CSO1, holds promise for not only sort of addressing the antithrombotic, but also reversing, which is required, the remodeling, which is so important. So we hope that we are addressing a completely new entity, which so far has not been addressed adequately in the pulmonary arterial hypertension therapeutic scene.
Yeah, so that makes sense to my next question. It was more CS1 is targeting PAH, and it's really rare and a progressive disease. It's so difficult to diagnose and treat. So I was kind of curious why you started with that indication.
I first addressed your question about difficult to diagnose. That's of course a clinical problem, which is not really part of our endeavor in PAH, because we are targeting patients that are already treated on stable medication, and we had our CS1 on top of that. But the reason why PAH is difficult to find is that in the clinic, the symptoms are very similar to heart failure, very similar to lung diseases. I mean, it's very difficult. And the diagnosis is an invasive method. That means that you need to do a right heart catheterization to have the diagnosis. And you don't start with that in a patient with dyspnea or functional disability or something. You start with X-rays and CTs and all kinds of different methods to try to rule out heart disease and lung disease, and then you kind of end up with that you didn't find the reason there, and then ultimately you do a right heart catheterization, and that is a process of maybe a year or two before you end up there. So therefore it's difficult to diagnose. But as I said, in our approach to the disease, we are approaching established patients already with a diagnosis. They are treated, but inadequately treated. They still have symptoms, they are functional disability, they have a poor quality of life and a poor life expectancy. We want to change that. And so we add it on top of established stable medication. That is our approach in the studies we are doing and plan to do in the future.
So, Rahul, this might be a good one for you. I mean, what does Cereno's lead program look like versus the existing drugs in PAH and I guess CS014 in thrombosis? I think you used to have leading positions at Bayer and Astra in these areas.
Yeah, no, thank you very much for that. And allow me to briefly just introduce myself. So by training, I am a cardiologist and emergency physician, trained in Germany and the US. I've had the pleasure of being in several big pharma companies and also biotech companies in senior executive roles. I've had the privilege of being involved in several launches, and in PH as well. So CS1, as I was saying previously, holds a lot of promise of not only addressing the vasodilation, aspect, which a few other drugs which are already on the market are, but we hold promise, and all the preclinical data is very promising in that as well, that we could also affect the remodeling, which, as I was saying before, is really decisive for the disease therapy and treatment. That is something important. To your second part of the question regarding CSO14, where are we going with that? Keep in mind, please, there we're of course alluding to what is the ideal position. And if one keeps in mind that the antithrombotic and other related drugs, besides their efficacy, however, also bring a bleeding risk. The data we have accumulated so far in a world-leading research lab at the University of Michigan holds a lot of promise that we may, besides the efficacy, also not have bleeding risk. We need to show that in patients, and we will be showing that, but the data is very promising, and we will pursue this direction further.
So can you explain, and Bjorn, you kind of touched on this earlier, can you explain some of the modifying characteristics of CS1 really in the context of PAH and how you're differentiated from the treatments that are available?
As Rahul mentioned, the existing therapies, they mainly are vasodilators, powerful as such, but still that's vasodilation. And if you discontinue that, it will go back. What we are aiming for, and that's what is called disease-modifying therapy. We think that we are in a position to have that. And that means that the therapy goes to the heart of the disease, the proliferation and fibrosis and inflammation that narrows the vessels. We think we can reverse that. We call it reverse remodeling. And with that, we can profoundly change the progression of the disease. And that means that we is something above or beyond vasodilation. And that we think will and could change both in a positive sense patients' quality of life and quantity of life. And this can be compared to actually the proliferation and what happens in the vessel to more or less a cancerous disease. And you know that HDAC inhibitors have been used in cancer and they have been successful there. And the similarities exist with the proliferation and what happens in the vessel. And if that can be reversed, we think we have this kind of holy grail disease-modifying therapy.
So HDAC inhibition exhibits highly relevant properties, which are really desperately needed, not only the cardiovascular arena, like anti-fibrotic properties, anti-proliferative properties, or antithrombotic, but also in the PAH arena. We have seen that while there has been a lot of progress been done over the last years, but it was mainly in the vasodilation field, not so much in reversing the disease, which our compound CS1 holds promise of. So we are very excited that we are bringing a different kind of approach to the disease of pulmonary arterial hypertension, an area where patients desperately need further improvement in their therapy.
So looking forward for CS1 phase 2, the readouts coming up, especially with the collaboration with Abbott for their CardioMEMS device, can you help us understand what clinically meaningful might look like?
Yeah, I mean, CardioMEMS is really a cool device. It's something that has been around for, I think, almost 10 years now. It has been developed and very thoroughly documented in heart failure, left heart failure mainly. It has been shown in numerous studies to be very safe. It has shown to reduce hospitalizations, and if you put all the studies together, it also can affect the mortality by the data that this can send to the treating physician on a daily basis if you want. So what you do with CardioMEMS, and that we use in our study, is that you insert in the pulmonary artery a small implanted device which is self-sustained, gets the energy from the blood movements, and it's not more than larger than a coin, and it can be interrogated how often you want on a daily basis and sent to a repository to evaluate by the physician whenever he wants. It can be daily, it can be weekly, it can be accumulated for a longer time. And patients have had this up till 14 years and without any malfunction, and the patient that had it for 14 years died for other reasons. So it was not that that device failed. So we think with this kind of device, we can monitor hemodynamics of the patients every day and look at the area under the curve, the burden of the heart, the function of the heart on a daily basis, instead of what you do with right heart catheterization, that is one shot sometime and then maybe a shot 3 or 6 months later. That's 2 or 3 observations. Here we have observations on a daily basis.
If I may add to that, Soo, the big advantage is, and Bjorn was alluding to that, and I had been doing right heart catheters, one has an insight in the hemodynamics, which are so important to optimize therapy just once, because it's an invasive procedure. CardioMEMS, once installed, gives you continuous non-invasive information, but hemodynamic information, which then can help optimize the therapy for the patient. So it eases the work of the physician, and it very much can improve the lives of patients because of this continuous information that one receives from this device. And we're unique in this, and in this that we're doing this in the trial. So this also sets us apart from all the other trials in pulmonary arterial hypertension.
Yes, I just wanted to continue on that foot, that this is a unique collaboration with Abbott. Abbott has not done any formal documentation of this device in PAH, but they want to, and they want to document this together with us. And FDA have actually not done this kind of approval for a study with the device and a drug at the same time. So both the departments, the drug division and the tech division of FDA, have together approved one IND for kind of both of these. And so this is a unique collaboration with Abbott.
Yeah, I mean, it seems like a great situation where you have a lot more data, and it's great for Abbott also. Beyond CS1, you have some preclinical candidates coming up that seem pretty promising. Could you recap those and then give us an idea of what the current outlook in reaching the clinic would be?
Yeah, we have 2 other assets which are preclinical. And one is CSO14, which is an HDAC inhibitor derived from VPA, but it's a new chemical entity. It has been thoroughly investigated in the preclinical sense. It has been shown in 3 different models to be very effectively antithrombotic without bleeding. It has gone through a full tox program and with flying colors, and it is now almost as we speak, but in a couple of months, going into first time in man. And we want to develop that for a thrombosis indication. That is the first asset. The other asset is called CS585. It's a prostacyclin IP receptor agonist, which is much more cell selective and long acting and potent than existing ones. We have not really decided the exact indication for that yet. We are still in the preclinical phase and documenting. But it has shown data as well, being antithrombotic, antiplatelet mainly, without the risk of bleeding. So we have two very, very interesting assets. One is now we pursue for thrombosis. The other one we have not yet decided, and it's a little bit further back. It's maybe going into man in a year or something.
That's great. This has been actually super helpful, Bjorn and Rahul. I'm really looking forward to seeing more data, and thank you all for joining us here today. If you'd like to learn more about Cereno Scientific, please refer to edisongroup.com. Thank you.